Okay, so thanks for the introduction. I'm Halvor, a PhD student here at Simla, and I will be presenting some work in collaboration with Ada, Marie, Miro, and Natalie at NTNU. So first of all, what are modal cilia? This is a cartoon showing our brain or caricature of our brain, and you have this squishy part that you see in movies. This is the brain parenchyma, or it's part of it, and inside it you have ventricles, which are hollow structures filled with cerebrospinal fluid, and lining the inner walls of all of the ventricles, not only here at the choroid plexus, you have these hair-like type of structures called cilia. These are have muscles and beat back and forth and contribute to the flow pattern inside the brain ventricles. This is a video produced by Natalie's group in a previous paper, where they have imaged the cilia on the inside of the brain ventricles of a zebrafish. So this black thing here, that's the hollow part with the cerebrospinal fluid, and then these are the walls of the ventricles. And you can see these cilia are flapping back and forth. What they also observed inside the same ventricles is this type of flow pattern, where you have some particles stained in a green color, and then the magenta is just what was there from before. So you can see, just to show the flow patterns more clearly, and you can see that there's like this type of vortex or rotational flow pattern inside the brain ventricles. What they further observe is that if you don't have the cilia, so this is still with the cilia, but without the heartbeat, which was making the flow kind of beat back and forth like this. What they observe is that if you ablate the gene expression of the cilia, you don't get any type of rotational structure. So without the cilia inside the brain ventricles, the whole flow pattern just stagnates. And this has been seen to be very important for the development of the brain of the zebrafish. So they develop neural pathologies in more than 90% of the cases. So we want to help Natalie's group better understand what are the underlying mechanisms of this cilia-mediated flow, and also link this to the transport that this flow aids in, because their working hypothesis is that the reason that the neural underdevelopment happens is that certain types of proteins or molecules that are important for the brain development aren't transported around in the way they should. So to investigate this, we have developed a computational model and calibrated this with experimental data of zebrafish. So using a surface geometry that Natalie's group produced, where you can see in magenta, that's where they've seen genes that express cilia. So we produced a computational mesh from this surface geometry. And then to model the flow of the cerebrospinal fluid, we used the time-dependent Stokes equations, justified by the very small length scale of these ventricles. It's about hundreds of microns in size. And then we couple this flow to an advection diffusion equation, where the flux is the advective flux from the cerebrospinal fluid and a diffusive flux. So to model the cilia, we have marked certain regions of the mesh where they see cilia on the surface geometries. And on these parts of the boundary, we apply a tangential traction to simulate the force of the cilia kind of dragging along the fluid. On the rest of the boundary, we just let the fluid slip freely. So this is motivated by the fact that in experiments, they they see the velocity profile increasing towards the wall. So a slip condition seemed more appropriate to model that type of behavior. And in the normal direction, we just impose no normal flow to model the ventricular compartment as closed. To simulate this back and forth beating motion that we saw in the videos, we take two ends of the ventricles and put normal pressure boundary condition. On one side, we just set 0, on the other, a sinus function to generate a pressure difference. Both of these, both the stress and the pressure are handled weakly in the Stokes equations. And then to model the transport, we look at 3 different injection sites on the ventricular surface where we set a Dirichlet boundary condition so that and initialize the rest of the domain as 0 concentration. Setting 1 on this part generates an influx of mass, and we do this weekly because we employ a discontinuous Galerkin method to discretize the equations. Since we want to model the compartment as closed, we would like to just set this j dot n equal to 0 on all of the rest of the boundary, but when we use these pressure boundary conditions, we've kind of opened up the geometry with holes where we get in and outflow of CSF. So we have to handle this in some way. And on the parts where stuff is flowing out, we just set a 0 diffusive flux. So, and then leave the advective flux in the weak form. And on the part where things are flowing, the fluid is flowing in, we have an approximate periodic boundary condition where we just say that what's flowing in is what flowed out at the previous time step in the lack of a better approach. We discretize the Stokes equations with a non-conforming finite element method. We use Bretzi-Douglas-Marini elements and for the velocity and DG elements for the pressure. So this was motivated by the fact that we were having troubles with the transport simulations. That's where the root cause seemed to be that mass was not conserved. So first we're using Taylor-Hood elements doing some tests on the zebrafish mesh. This is the max velocity of the divergence when using P2, P1 elements, and this is the L2 norm, which is clearly not 0. And this is when using the BDM DG approach, which is in the HDiv space, and when you have these discontinuous pressure elements, you get exact mass conservation. And also, when implementing this slip boundary condition, you kind of get this normal velocity equal to 0 for free, because the BDM elements have their degrees of freedom associated with integral moments of the normal component of your variable. Whereas with the Taylor-Hood, you would have to do something more fancy with, for example, a Lagrange multiplier or a Nietzsche method. So for the transport equations, we use discontinuous Galerkin elements and also upwinding because we have large Peclet numbers inside the domain. We numerically verified the model, seeing convergence on a cylinder test mesh. We also see consistent results on refinement of the zebrafish ventricles mesh, and also the very low divergence that we want. So how do cilia affect flow patterns and transport rates? We can start by seeing this video that we saw earlier, where you see the velocity field is kind of rotational with some beating back and forth. And using our model, we get kind of the same structure where you have these rotational structures in the compartments where you have, so we have cilia up here, down here, and here. and also this beating back and forth because of the pressure boundary condition. And we compared transport in the ventricles for different types of molecules by considering a range of diffusion coefficients. So we considered we have the diffusion coefficients of 4 different proteins, and then we set like the smallest is the lower diffusion coefficient, the highest is the higher, and then we have one in the middle. And then we compared the transport for the 3 different ones. So in all of these, this very dark green part is the injection site. And then this is with the smallest diffusion coefficient, a medium one, and a larger one. So we see that for the very small diffusion coefficient, it's really, one can really see like the same type of flow structures as in the velocity field, whereas it's more smudged out for the higher diffusion coefficients. We also looked at how the transport is when considering different injection sites. So in this case, we took one diffusion coefficient, and then we considered the 3 different injection sites, which I showed earlier. So one on the anterior part, one in the middle, and one on the posterior part. And one can see that the transport is really governed by the solute actually reaching these cilia regions to kind of be transported along those routes and then further into the other parts. Whereas here, where we don't have any cilia, it's really just diffusion governing the whole transport process. So the current outlook on this work is that we will compare this with some experiments that Natalie's group are performing now to see how the transport we simulate compares with the ones that they see in experiments. Maybe some further analysis of the discretizations and these problems with when not having exact mass conservation, and also try to apply this to the model which we have benchmarked to this geometry, which was 2 days post-fertilization of an embryo. So you can see this is from day 1 to day 7 of the life of a zebrafish embryo. The brain ventricles really develop in structure, so the transport will probably be very different for all of these geometries. And then the most important things last, thanks to all collaborators on this project. Thank you.